In SomaSensing, we guide you to your sensory awareness, your inner felt sense, to unlock your ability to self-regulate. And one of the ways that we tune into the body is through our body's fascia. And in this video, I want to share with you a lecture that was given by Robert Schleip, one of my mentors. He's a fascia researcher and body worker, and he talks about fascia as a sensory organ, or fascia as a sixth sense. And the way that we have this conversation is from my own curiosity. So please dive in. I hope you enjoy this session as much as I did, especially if you are wanting to go down the route of somatic education. Hello, everyone. I'm here with one of my favorite fascia researchers, Robert Schleip. Robert is the director of the Fascia Research Group at Ulm University. He is a pioneer in fascia research and has been sharing a lot of his work over the years. When Robert and I met, it was, well, not met, we connected in 2011. And what I appreciated is Robert's generosity in sharing the research so we as somatic movement therapists are better informed. So I'm really delighted that he's agreed to do a session with us on fascia as a sensory tissue. Oh, and I must add, if you are familiar with the Fascia Research Congress, Robert was instrumental in bringing that together in 2007. So make the most of this, because it's really going to help you to understand the sensory wisdom of our body. So Robert, I'm going to let you dive in. And if there's anything else you'd like to add about your background or research or anything exciting that you're doing for us.
Well, thank you very much, Yasmin. I respect your work very much. We have been in dialogue. I have a great appreciation, not only for your curiosity and following our research field for such a long time, and for asking such curious questions, but also for the nature of your therapeutic work, in which I have some background, not as profound as yours, in my past in having worked with people like Emily Conrad, the inventor or co-inventor of the continuum movement method, the Hakomi method in terms of body-oriented psychotherapy, Peter Levine from somatic experiencing, and maybe today also coming to Stephen Porges. So I have worked with him. And you asked me about to share now with some slides, and maybe that's how we get started. And then probably we go into an open dialogue afterwards. So what has been new in the field of fascia research in the last couple of months or years? So let me go to this presentation and share my screen. Just get back. Screen sharing. It should be this. There we go. Okay. So, Yasmin, you should be seeing a slide with Fascia Nomenclature Committee on the upper left. Wonderful.
Yes.
So this is working. So when I talk about fascia now, I'm no longer using the established anatomical definition of fascia, although that is still in usage and is still recommended when you talk with people in the medical allopathic school medicine establishment. In their usage of the term fascia, which is defined by the Terminologia Anatomica, which is still in use, but it's 17 years old that the last version has been revised. Their fascia are only dense, sheet-like layers of connective tissues that you can cut with a scalpel. So it applies to the fascia lata on the outside of the thigh, that is usually much stronger than on the inside. It applies to the lumbar fascia, to the nuchal fascia, maybe also to the plantar fascia on the bottom of the foot. But even there is a question, because the fascia on the bottom of the foot most of the collagen fibers are in parallel. And then the classical terminology would say this is a tendon sheet, an aponeurosis, and they made a distinction. If it is in one direction, they called it a regular connective tissue, and fascia was considered to be irregular. And so there is also some discussion in the medical establishment which tissues to include and which not. But in our field, that has been influenced by more broad thinking. That has been started, not really started, but propagated forward with the fascia research congresses happening every 3 years. And the first one in 2007 that happened at the conference center of Harvard Medical School, where we had been greatly influenced by the tensegrity model, in which stability in a biological organism, and humans are now simulating that in the way how they do bridges and buildings, by the interconnected tensional network and not so much by the posts holding it. And therefore, the new definition of fascia is a body-wide network of collagenous connective tissues, no matter whether it's sheet-like, whether it's ellipsoid or round. You could call it a tendon, you can call it a tendon sheet. They are all local expressions of one body-wide fibrous connective tissue arrangement. So that is the term of connective tissue that most, but not everybody, is using recently. Some people are now using fascia synonymous with connective tissue, and they include bones and even blood into it. And that's also reasonable to do. And some people like the name fascia so much that they include every mechanosensory tissue in the human body. That means everything in the human body is for them fascia. But the majority of the people who are inspired on fascia are following now this more functional definition, in which all fibrous collagenous connective tissues, whether they are around a muscle and you can cut them with a scalpel, or whether they are very small, like the little pockets around a little single muscle fiber, you cannot cut it with a conventional scalpel, you need a microscope. That is now also included as connective tissue. So that is a clarification I had, particularly when I now look at the innervation from the sympathetic nervous system. And there has been some advances in the last year, and they have been not only looking at the epimysium, but also the loose connective tissue under the skin that we call superficial fascia. And in conventional anatomy, they didn't call it superficial fascia all the time because it was not dense enough. But for us, it is part of that. But also the loose connective tissue in the ear within the muscle. And you see here on the left, these dark brown fibers are sympathetic fibers. And the sympathetic nerves belong to the autonomic nervous system, and the sympathetic branches of that go almost everywhere. So they are spread wherever you have any kind of muscular or organ tissue, because wherever you have blood vessels, you also have sympathetic nerves. And most of the sympathetic nerves that they found in fascia are actually, they have a lumen. So you can see they are around a longitudinal vessel. And then we think we know what they do because they are vasoconstrictors. So they regulate how warm your hands are getting, how you're blushing, how hot your head gets when you are excited, or when you are under certain emotional stances, conditions, you may get cold feet, etc. And you also see that in the picture on the very right from Professor Neuhuber. These red nerves are actually around a blood vessel, but you also have some sympathetic nerves, here colored in yellow, that are outside of a blood vessel that you see with a double arrow there. And there we don't know what their function is. They have a function separate from vasoconstriction. And now the speculation is that maybe they have a trophic function, trophic with pH, that means a nutritional or metabolic function, that they may change the chemical condition in terms of a pro or anti-inflammatory direction in that local tissue. So we know fascia is very densely innervated. Actually, the sympathetic innervation is the most densest. If I go back, I can show you again. This is the latest publication from Siegfried Menze, just published a few months ago, where he said 40% of the entire fascia innervation, including proprioceptive innervation, including nociceptive innervation, so many different nerve types, but the sympathetic nerves are by far the most frequently. Very interesting that some of the muscular connective tissues have a higher sympathetic nervous system innervation than others. And these are these red colored tissues on the back of the body, as opposed to the yellow or green colored tissues that are more ventral or more to the side. And that has to do from which branch coming here from the spinal cord are you innervated. So out of the spinal cord, you have the spinal roots coming down, coming to the sides, and they are innervating. Like out of the lower lumbar spine, you have the innervation of the whole leg going down. And so if you look at these spinal roots, there is one branch going pretty soon backwards, and that is the dorsal branch. And that innervates these red colored tissues, and they have a much higher influence from the autonomic nervous system than the tissues that are innervated from a lateral branch or from the regular ventral ramus here that comes out of this basic branch here. So that is a good explanation. When people are under stress and you get the shivers, or you get sweat, or your hair sticks up in animals, so when they are frightened, it is the hair on the back, not so much the hair in the front or in the face, that is getting into a stress response. Or if you have the shivers, that where something dangerous happens, or you think it's happening, and a second later you realize, oh, it went well. So these shivers do affect the posterior tissues more. And so these examinations that I showed before, they have been done with the thoracolumbar fascia. So the thoracolumbar fascia seems to have a more profound innervation by the sympathetic nervous system that you have there. And then the question is, if you are under chronic emotional stress, that would be chronic sympathetic activation, or one style of chronic sympathetic activation, does that change in any way the sensitivity of your myofascial tissues? And they have examined that in animals, and you can discuss why you need to do that with animals, but they did only a mild torture. Let's discuss whether that is torture, because we often do it to our own children and to our own bodies as well, where you put the animal or your own body into a plastic container, and you say, sit still for the next hour. Don't move, don't bounce around. So if you go into the opera and you sit in the 8th row, it would be socially irresponsible if you get up and dance, because the people behind you cannot see it. So you force yourself to sit more or less still for 1 hour. And that's what they did here with the plastic container. So the animal can breathe freely, but it cannot change its body position for 1 hour. And you would say that is not torture. However, when you do that for 1 hour per day for 12 consecutive days, you see some changes in the nervous system condition of that. The first thing is that they found out that the allocation of receptive fields, so when you touch or stimulate a given fascial or muscle tissue, and you measure which areas of the spinal cord are stimulated, normally you have a very much segmental innervation, where one muscle is innervated from one level of the spinal cord, and you have these dermatomes or myotomes. They are quite valid if you have a non-stressed, healthy, pain-free German client, or no, so not German, but normal client in front of you, or an animal. However, if the person is in pathological conditions, here under chronic emotional stress, then the beautiful maps that you may have seen in the books are often more blurred, because then one muscle is influenced from 3 different segments. So that was a nice surprise and explains a lot. So under chronic emotional stress, afterwards, one muscle is innervated from several spinal cords, and you have probably more referred pain. So if you have an irritation in one tissue, neighboring tissues will be affected as well. What they also found out.
So Robert, can I just step, kind of dive in here? I wanted to, there's 2 questions that I have.
Yeah.
One is, you talked about fascia and the fibers of fascia. And in this kind of new way that we're looking at fascia, where does what I call the gel, or what's been called the extracellular matrix, and I describe it to my students as the saliva in your mouth, for example, which can be everywhere in your body. Where does that fit in with our fascial description?
So in the more functional definition, that would be included. So so the loose connective tissues, for example, the superficial fascia under the skin. Is more saliva-like.
Okay.
In animals, that layer is quite thin, except in pigs. But in humans, this kind of sticky, gluey, half-wet, viscous layer is not dense sheets. It's not as, you cannot cut it very clearly. So it's half liquid. And that layer is
so I usually describe it as a gel, like a gel, if I'm from a somatic perspective. It's like, so, you know, that the fibers are bathed in a gel, for example, or if there's no empty spaces in the body, there's this areola, what we used to call areola tissue. It's the gel, right, within the body. So that was the one question.
So that's included in the functional definition now.
Yeah, so that would be in there. That's great, because often when I'm describing from a movement perspective, I would talk about tuning into the body as a container, and can you imagine that the body has this gel? How stiff is the gel? So that people have something to feel, and then maybe just move. Like you said, if you're sitting for long periods of time, the body doesn't like to be still. It might move a little, you know, it's just like that. And that's what we encourage in intuitive movement.
Very interesting. Before you come up with the next great question, this gel-like layer is mostly under the skin, and it's thicker there than in cats, horses, dogs, etc., in humans, and also around our visceral organs. So that's where we have this viscous layer more.
Yeah, and it allows the body to spiral. You spoke about under the skin as well. So one of the things I'm finding with motion, there's a movement that I call a skin gliding motion.
Nice.
Yeah, so, you know, on the floor, sending the floor away, and the whole fascial net is moving, right? And you know, when you send the floor away, the entire net moves.
And they are usually separated. So you have this viscous layer on the periphery under the skin, and that would be more of these external movements. But in front of the neck, this viscous layer under the skin is connected with the visible connective tissue that surrounds your heart, that is inside of your belly. So they are the two systems, the inner viscous ocean and the outer are very much connected.
Yeah, so there's this continuum. And it's interesting because you were talking about the sympathetic innervation in the dorsal or the back of the body, where you have the thoracolumbar fascia, you know, you have dense tissue. I just call it the upper body, the neck and shoulders and lower back, where people often describe stiffness, don't they? So when they come, it's like, oh, I'm stiff in my neck and I'm stiff in my lower back. So we start to do these movements. But the question I have about that is, you talked about sympathetic innervation, and you mentioned muscle. And I often, these days, I'm just saying fascia, because of the interpenetration of the fibers with them. It's almost like the muscle cells are shaped by the fascia, or you know. So I've started to talk more from your session the last time, how fascia is highly innervated and more so than muscle.
Yeah, sure. Yeah.
So
if you give the muscle spindles as the sensitive innervation, then the innervation of the intra and extramuscular fascia is 6 times higher, not 10 times as we originally thought.
So I thought 7 times was the last time we heard. 7 times. So 6 times, so it would be the epimysium, the endomysium, those layers. Okay, and there's also kind of this gel in between. So a third question that's popped up is that if we have this other gel within the body, so we've got the thoracolumbar fascia, some of the areas that I find their stiffness is in the diaphragm. So could that gel become stiff too within the body from a sympathetic state? Okay.
It can become stiff on 2 different ways. The first one is that the gel develops a higher stiffness, similar like a honey that's resting for years without being stirred. It develops certain crystals inside, and then if you stir the honey pot, it has more resistance, so it has a higher viscosity. You would also get it if you take ketchup and you put it in the freezer. So then the same material doesn't change its composition, but the chemical condition becomes more harder. And that would be a change in the ground substance, but not in the collagen fibers. And that can happen quite rapidly, and you can also break it up by applying heat and by stirring the honey. So you may loosen up some cross links, some attachments, some adhesions between collagen fibers, but you don't change the collagen or rupture the collagen fibers. So that would be easy. But if you have that over years, then you may have a fibrosis of the fibers. And that is much more difficult to loosen. So that is something where we hope to have the ultrasound in the future refined enough that you can find out if the sternocleido in a person that has been stressed for years, you can often see it, that this muscle here becomes like a steel cable, and you touch it and you say, holy cow. Especially your right leg.
That was me. When I did lots of Pilates, I had this massive sternocleido muscle on either side. But that's fascinating because that really fits in with the intuitive movement, the way that fascia revitalizes through those coaxing movements. And you spoke about the thoracolumbar fascia. So I'm going to dive in again. So I'm going to let you continue because I just wanted to make that distinction between the gel and also you talked about muscle innervation. And I'm trying to stay away a little bit from the muscle innervation because people talk about muscles contracting and releasing and they forget about the fascia. So it's just kind of diving in. So I'm going to let you continue.
Thank you, but this is nice. So let me see. Here I am. So this is the animal, but it could be you, your children, many people you know, that had mild, moderate emotional constraints for 12 consecutive days. And what they find out is that the pressure pain sensitivity is much higher, or their threshold for pressure pain is lower. So if you push anywhere in their body and you start with very mild pressure and then you push gradually stronger and stronger, eventually at some point you will say that's too much. And they found out, so if you press with 50 grams only, almost no animal will complain. But if you press with 5 kilograms, most of them will make a avoidance behavior. So the threshold is much lower if in the previous 1.5 weeks you had this repetitive emotional strain there. And that is very interesting. So, and that is not on one place of the body only, but it's all over your body. So there could be one reason, but there are also additional reasons, that somebody is pressure sensitive all over their body, particularly since they had this stress period in the last 2 or 3 years at work, in their marriage, etc. An additional one is called repetitive stimulation. So if you do 10 times potentially irritating stimulation, so you push them 10 times or you tweeze them 10 times, that is always experienced as more agonizing than only one time. But in a normal person, if I push you here 10 times, you would say that's maybe twice as disturbing as pushing me only once in terms of the magnitude. But in some people, it escalates. So if you hear noise and you hear it again, then finally it drives you crazy, but it's the same size of the noise. And that wind-up phenomenon, or the sensitivity for repetitive stimulation, that is aggravated when you had this chronic stress stimulation previously to that. Now that is animal research, but humans should be probably different. So there is now human research also, where they looked at people with chronic low back pain, and they asked them, did you also have childhood post-traumatic stress disorder based on childhood abuse? So that is one type. There are different types of post-traumatic stress disorders where you have most research. Depending on your definition of childhood abuse, whether you only look at sexual abuse or whether you also include emotional abuse, it varies in our Western population between 18 and 51%. And we have questionnaires to evaluate that. And what they found out is that those people who not only have low back pain at the present time, but who also decades again report about a previous time in their life where they had chronic emotional stress due to childhood abuse, their sensitivity is also different in the same way, that they have a lower pressure point threshold, very, very interesting, than people who didn't have that and only have the current low back pain. And in addition, so that's how they do the wind-up phenomenon. So there is a little pinprick needle and it irritates you once. It's not very painful. But if it does it 10 times, it irritates you much more. And if you had a period in your life, decades ago, where you had chronic emotional stress, then this irritates you more. So that probably tells us that the sensitivity in our myofascial tissues can be influenced by chronic sympathetic activation. And we need to know more about that. And this is now a happy time, Yasmin, for our group at Ulm University, because we have been researching the influence of the sympathetic nervous system on tissue stiffness in fascia, not in muscles, but in fascia, for the last 10 years. And now finally, we were able to publish our research last year. And it took us a long time because we wanted to publish it in a high-ranking journal. We could have published 3 or 4 years before, We had the results there, but we wanted to have it published in a high-ranking journal so that it's really being accepted and taken care of and criticized and built on by other researchers worldwide. And that finally happened in a very nice way. So this is now very well published. And we looked at the population of the density of myofibroblasts in fascial tissues, because our research was inspired by the old German phlebology professor Staubesand, who had also found a high sympathetic innervation in fascia, but he reported the presence of smooth muscle-like cells in fascia. And he claimed that all the smooth muscle-like cells in fascia would be innervated by the sympathetic nervous system, and they would make the fascia stiffer. So we looked for the smooth muscle cells. We didn't find smooth muscle cells, but we found smooth muscle-like cells, and they are called myofibroblasts. They are known from wound contracture, for example, but we found them also in normal, non-pathological tissues, but to different degrees. And we found out that in the human thoracolumbar fascia, you have a much higher density than in the fascia lata of humans, these dark brown cells, or in the thoracolumbar fascia from animals, for example. And we also showed that the contractile quality of the tissue, how strong it can contract, if you stimulate it chemically, the magnitude of contraction is associated with the density of these cells. So that makes it more likely that actually fascia is a contractile organ and that the contractility depends on these smooth muscle-like cells. And we thought they were innervated by the sympathetic nervous system originally, but when we tested them with different substances, we couldn't find a substance that's associated with the autonomic nervous system first. We only found substances that have to do with wound healing. For example, TGF-beta or histamine-like substances. But we were able, in an organ bath, to have fascia actively contract. But the question is, how strong can fascia contract? And we did a calculation if all the fascial tissues at the cross-section through your lower belly, and if all the red colors, spinal stabilizing muscles, if their fascia contracts all together, that would come to 1 Newton, that is like 100 grams only. That is enough force to lift your finger, but it's not enough force to lift a box of water off the ground. So it's not very impressive. And that was a disappointment for my friends from the martial art field, where they still have the question and they were hoping to find an answer. How can people who are not very muscular, they are not like a bodybuilder, have so much power like Bruce Lee was as a martial artist, supposed to be very, very strong with his pushes. And his muscles cannot be those strong, so they speculated it must be the fascia. But if you calculate how much the fascia can contract due to the myofibroblast, and you calculate the maximal force, you realize it can only be 100 grams if you look at a small time period. So that was a disappointment for them. But if you give a longer time period, then you not only have tissue contraction, but contracture. And that's what you see here. This is a cell, a contractile cell, that within 1 hour travels through the collagen net in a left downward direction. So it started here, and 1 hour later, it's in the middle of the picture, and it leaves behind a densified collagen net. So that is now the model that we have. If you are under stress for a long time, then your myofibroblasts are traveling through, probably while you rest, and looking for places where they repair the connective tissue. And if you then have strong stress-associated cytokines or hormones in your body, then they will do that with a stronger contracture force. And then maybe after 20 days of that, you may wake up in the morning feeling that your underwear, not the underwear outside of the skin, but under the skin, has been in a dryer with too much heat, so it has shrunk. So these cells are ridiculous in terms of how much force they can exert within 1 or 3 minutes. But if you give them 20 days, they can tighten your collagen net to such a degree as you have in frozen shoulder. And frozen shoulder is driven by these cells. So we also found some people where the density of these cells is comparable to frozen shoulder in the lumbar fascia. So maybe you have frozen lumbar fascia in some people, but they wouldn't get that in 3 minutes, but they could get that in 3 weeks via these contractile cells. But first we were disappointed because these cells were not stimulated by any autonomic nervous system transmitters until we found a new paper in the field of psychoneuroimmunology. That is a very interesting field, that they know when people are under chronic stress activation, that their immune system, very interesting now in the corona period. So how is your immune system strengthened and sometimes weakened in the way how you fight against possible infections? And that was known that when you're under chronic stress, you have different T3 cells in your lymph node, but the transmitter was not known. And they finally found the transmitter in a recent paper. And that is one of the substances that we used in our laboratory, that we didn't associate with the autonomic nervous system. And now we have an axis, so that transmitter called transforming growth factor, TGF-beta1, is well known in wound contracture studies, but now it's also associated with chronic sympathetic activation. So now we have a link, and also in addition, in cell culture, not in our organ bars, they showed that the fibroblasts in connective tissue have alpha adrenergic receptors on their cell membrane. And if that is stimulated by adrenaline, that increases the TGF-beta production. And then you have an indirect link. So it's not that you have sympathetic branches synapsing with the myofibroblast. That was our original model. No, but the sympathetic nervous system leads to an increased expression of adrenaline, for example, and that stimulates the myofibroblast to produce more TGF-beta. And if then more TGF-beta is in the area, that works back on the myofibroblast to become more contractile and to make the fascia more stiff. Now, so we know that the sympathetic nervous system is very important to change fascial stiffness. If somebody has a real stiff sternocleidomastoid, and it's not only the muscle tonus, but it's the fascial envelope, then that could be one of the backgrounds.
So can I dive in here, Robert,
Yeah.
again? Because this is really fascinating for me when you mention psychoneuroimmunology. In 2010, I did a course with a neurosurgeon in South Africa. His name is Dr. Ian Weinberg. And he was running workshops on psychoneuroendocrinology.
Mm-hmm. That's exactly the field. Yeah.
Yeah. So I was familiar with that just before we met, and I wondered how they fitted together. So now that you're saying this, it's brilliant, because I was trying to find how does it link in with the chemistry change of the tissue, right? Mm-hmm. And he mentioned interleukin 1 and interleukin 6, and tumor necrosing factor as well was the other. So I wasn't familiar with TGF, so that's new for me.
Yeah, was new for me. So these interleukins, they are part of the inflammasome, so pro-inflammatory.
Pro-inflammatory, yeah.
And there is a lot of research there, but that was influencing the nervous system. It was influencing also the muscle sensitivity, but it didn't have a direct connection to fascial stiffness. And the TGF-beta is in an interdependent relationship with the inflammatory cytokines. It's also a cytokine, but it's not per se a pro-inflammatory one. But they are influencing each other. So and the TGF-beta influences the stiffness. And there we have the link. Yeah.
Well, that's so interesting for me, because I never had that link. So that distinction that you've just made is really important for people to understand, because, you know, there's so much with social media, you know, one person will say this and they'll read a paper. And I think it's so important to have that distinction from fascial research than just to make assumptions that the issue is in the tissue, right? But how is it in the tissue? Something else that I wanted to, what's really come up for me is I had just said to someone yesterday that fascia doesn't contract. So I have to take that back and go back and say fascia does contract, but the amount that it contracts is not like muscle, right?
Yeah, you wouldn't be able to see the contraction if you stare at it. But if you come back after 3 weeks and move the shoulder again and it's stiffer than before, then you can say, holy cow, this is impressive.
Yeah. Yeah, so it's got this
contraction in terms of a short-term process, no. But you say it's able to stiffen over time. And then you don't call it contraction, you call it contracture. So it includes a remodeling of the stiffness of the ground substance and the fibrous matrix.
Okay, so that's also another distinction, because it's the fascial contracture. It's feeling stiff, but it's because of the contractile nature of the tissue. Okay, so I've got that. So thank you for that.
Also, you helped me to clarify this. The rapid pro-inflammatory changes that you have if you have an injury, but also if you are under chronic stress or if you have an infection, they don't change the fibers immediately, but they change the ground substance. For example, in pro-inflammatory conditions, you may have an increased hydration in terms of tissue swelling. And then later it makes the ground substance more gluey. So connective tissue is what an engineer would call a composite. A composite is the way how you do bridges, where you have a fibrous matrix surrounded with an amorphous mass of concrete, etc. And nature does the same thing with connective tissue. So you have collagen fiber bundles in one direction or in several directions, and you have the sticky glue around it. And that is much more sturdy against being ruptured than only one of these two compounds. So you have the viscous amorphous mass, that's the ground substance, and that changes more rapidly with pro and anti-inflammatory cytokines. And you can influence that more rapidly with movement and also with heat application and with stirring the honey. But the TGF is not changing the ground substance so much. It's influenced by the ground substance, but it makes the cables different. And it spits out more cables so that more cables are in the bridge, so that the bridge doesn't tear so easily, but it also makes the bridge less flexible for movement. So that would be a nice distinction.
Yeah, that's really, that's I'm fascinated with that, because I'm wondering whether it's the same with scar tissue or same with scar tissue.
So the scar starts with a swelling. That's the pro-inflammatory cytokines.
Yes.
And then later you have fibrosis. So you have lots of collagen around it, like in burn scars. the fibrosis is more long-term and more detrimental than the original swelling.
I've experienced that actually because with a burn scar, I had a burn on my leg and it formed a keloid because my body's Viking body, like you described, right? But what was amazing was I had so the keloid was removed. I had a skin graft. But I was young. Was, you know, when you're younger, it heals a lot quicker. But it was movement that brought back the shaping of the tissue. And I remember going to a workshop with someone who had trained with Tom Myers many years ago in the UK. And, you know, we were looking at, I was kind of looking at the scar, and he said to me, the scar's healed really well, but it was through movement. So I just wanted to confirm, is it the same with scar tissue? Also, is it the same then? Because when we talk about stress and what happens in the body, there is also fibrosis that can happen around the heart as well. So it can, right?
And the same is now happening with the lung, but also other organs with the COVID infections. And a lot of what people die is not the original infection, but it's a fibrosis. And that is so bad that sometimes it squeezes the blood supply off, and then the body produces even more fibrosis. And so the big question is how you can stop the stress-driven fibrotic proliferation in the connective tissue that the body does as a defense, even if you kill yourself with it. So you did very well by bringing gentle movement into the area.
Yeah, that's what I'm working with. And the gentle movement based on the emerging intuitive movements like we do in somatic practice. Some people call it childhood development movements, but I call it emerging because they are rocking spontaneously. So that's really helped me to understand the distinction is so important, because I wasn't sure about the pro-inflammatory cytokines, and then there's TGF, and I'm so excited. So I'm going to let you carry on because I'm sure.
Okay, so let's come back. I'm getting better at sharing this. Yeah, very well. So this is a model that you and I have been influenced and very thankful, where we look at the autonomic nervous system, not only in the conventional way to say that the autonomic nervous system is like yin and yang, that you have the parasympathetic part that's good for relaxation and for digestion and for regeneration, and that the sympathetic nervous system would be for fight and flight. But there is also now a social nervous system as a third contribution from the autonomic nervous system. And that is now a model that's having more and more recognition in recent years. It's not been generally taken over by all the neurophysiologists, but more and more people follow it. And I'm very proud that I know the originator of that model, Stephen Porges, personally, and have been familiar with his model. And I can say for clinical practice, it's extremely valuable. In the neurophysiological field, there is some discussion whether the hierarchy is really that simple as he had originally proposed. He had proposed that in the evolution of animals, that the oldest fish only had one way to cope with threat, that would be by lowering their metabolism and oxygen consumption, that the parasympathetic nervous system would be the oldest, and that the sympathetic activation came later. That seems to be mostly true, but not necessarily, because usually nature and evolution is more complex. And he claimed that the social nervous system that I'm gonna explain is a new development that only mammals, and particularly Homo sapiens, and then we put ourselves above our animals. We are the most evoluted, and usually that's not the case. So it may be a bit too simple in terms of hierarchy, but in terms of clinical usefulness and understanding, it is very, very profound. And what he showed is that the vagal nervous system that used to be associated with parasympathetic activity only, that there are some branches that are innervated the face, the vocal muscles, and neck movements. And they belong to the vagus, to the parasympathetic nervous system in the conventional activation, but they have a different regulation behind it. And they are not necessarily into activating or into slowing down, as you can normally say for each muscle and each organ, what the sympathetic and the parasympathetic nervous system, they are into modulation, and they are related, and they are highly activated in humans, but not only, in social expression. And that is something that seems to be very important, and where you can recognize if you are under, or your mate is in a mild or moderate emotional stress, when their voice gets more monotonous. Hearing gets more sensitive for high-pitched irritating sounds that you have there. And you can see it in neck movements if somebody is talking with you. And that helped a lot. For example, in movement therapy or in massage, if you find the neck muscles activated, the sternocleidomastoid, the trapezius, these are often the first muscles. If you give them a relaxation, it then influences also your erector spinae, etc. So they seem to me more acutely mirrored with emotional activation that you have there. And also, if you get the client to do a breathing meditation, where they lower their breathing rate, and you do certain sounds with the lips, with the tongue, with the eye movements also. All these are one way of influencing the autonomic nervous system via what he calls the ventral part. So it's more here coming to the front of the body, or what he calls the social nervous system. So that is very helpful. So if you make soothing sounds, if you do chanting with people, if you do neck motions that are not into fight and flight, that can have a whole body-wide effect that we have a new explanation for it. When you have patients who are under pain, you may use the autonomic nervous system, but you can also use proprioception as a stimulation. And that is also a new explanation, because in the dorsal horn of the spinal cord, you have these WDR neurons, wide dynamic range neurons. And that means they are receptive either for nociceptive innervation, here the C fibers in red, or the blue fibers that are communicating indirectly with them that can signal proprioception. And some of you know the gate control theory from 10, 20 years ago, where they've said they are competing with each other, proprioception and nociception, from the same innervation area. Which one is faster at the spinal cord here? But the speed doesn't matter. That is a new insight. So gate control theory is no longer 100% valid, but it's partly valid with the new insight that we have now in recent years, that these WDR neurons, wide dynamic range neurons, or polymodal cells in the sensitive portion of the spinal cord, they are hungry for input. So if they don't get input from the periphery, from the area of innervation, let's say from the lateral side of the knee, or between two layers of fascia on your lower back, if there is never any segmental movement between the two layers, if the two layers always move together, then the WDR neurons that are linked with the connective tissue in between, they are not getting any stimulation for days. And that's not good for these orange cells. Then they get nervous and worried about their survival. And what they do then is they take out a magnifying glass and look for any potential nociception. If there is a slight pro-inflammatory cytokine in their area, then they don't measure movement, but they measure sensitive chemical threat in the area. So lack of proprioception can augment potential nociception. So that means proprioception and nociception are in a mutually inhibiting relationship. One lowers the other. So if you increase proprioception by giving the person more afferent input, then these WDR neurons are taking their magnifying glass down. You know some people that they have a leg length difference of 2 millimeters, and they have no problem, or they have 2 centimeters. But the sister has 1 millimeter difference, and she gets sciatic pain. Or you have a very mild deviation after your dentist did something where a hundredth of a millimeter in your jaw is not working, and you get tinnitus. You could now fix the last hundredths of a millimeter and get rid of the tinnitus, but then a week later, something else in their body is one millimeter off. It may be better to provide the jaw with singing proprioceptive stimulation, and then it doesn't matter if your jaw is a tiny bit more forward on the right than on the left. So this is a very profound inspiration. If you give the spinal cord from the same region, so if you have neck pain, it's not having a long-lasting effect if you wiggle your big toe. So if you have neck pain, you need to come with proprioception in the neck area or in the neighborhood of the neck. And then, not by the cortex, but by the spinal cord, you can shift the balance of competing proprioception and nociception. So that is quite a nice model. On the mechanical perspective, I want to share something which you probably feel also excited about, Yasmin.
Yeah.
That they found out when you stretch the Achilles tendon, that it gets wider in the right to left diameter. And that's not what I would have expected. If you take another tissue and you stretch it, you expect it to get more narrow in the middle. And then guess where would it be rupturing first? At the middle, of course, where it gets more and more narrow. And that would be the way how we humans would do longitudinal bundles. But nature does it in a very surprising way. So if you stretch the Achilles tendon, it flattens and gets broader. And that increases its rupture strength if it does that. And that's called auxetic behavior. So if you get long, and that's very important in dance. So if you want to get taller, many people are pulling in their belly in order that you feel like you have constant volume. If you want to get taller, you need to get more narrow down here. But there are dancers, if they go bigger, their lower ribs are going to the side and they spread at the same time. And that is a very different way that apparently nature is using quite often that you have there. And they now do that in nanomaterials, where they have materials when you stretch them. So the normal behavior would be down here, you have a material and you stretch it in this direction, then it gets narrow in the other direction. And that's what you, for example, have on the iliotibial band. So if you stretch it in one direction, and it helps the muscle actually to have more contraction force. However, in the tendons, if you stretch it in that direction, it gets broader. So the body is using it depending on what it needs more. And that is a very simple, well, I have a video, let's see if it plays here.
Yeah.
So this is what they are now using for medical material if they repair your valves. And you can see that this material gets broader if you stretch it, and it gets narrower when it's loose. And that's due to the wave-like undulation that you have there, that you have in ligaments. So we haven't figured it out yet at what body areas do you need auxetic behavior and where do you need the flattening behavior that is also useful. Yeah. For example, so when you have two directional arrangements in the epimysium of the belly of a muscle, it makes sense that if you pull it apart, it helps the muscle to contract in itself.
Yeah, can I just ask you a question about that? Because I think for me, in terms of movement, from kind of the fitness background and looking at muscle and how it moves, to this auxetic nature of the tissue has been a real fascination, because I didn't know this word auxetic until a couple of years ago. But what happened for me is exactly as you described with the dancer. Mm-hmm. So the first thing that happened was to stop people pulling in their core.
Yeah.
Right?
Yeah.
So that happened. That stopped a long time ago. But what began to emerge was this shape changing. So we started with the conversation around the body's not linear, but it can shape change. And what we describe as pandiculation, that urge to stretch and yawn, I find, as you've just shared, how the body has this efficiency, that every time the body moves, there's a mini sensation of what I call an expansion.
Yeah, but, expansional movement maybe, yeah.
Yes, expansional movement. So for example, if you pull the tummy in and you're reaching, it doesn't give you that, I can't even call it range. Yeah. But there's almost like, exactly as your video, and I have a similar video where when there's an expansion, it feels like you have this ability to move further.
Yeah.
But you're not actually stretching anything. It's just the spiral nature of the movement that allows you to go a bit further. So that's what's emerging from the intuitive movements. So I would say that my only thing is about the muscle and the flattening of it. I'm not familiar in the intuitive movement part, whether that comes in, because all I'm finding is in the spontaneous movement, there's the expansion. So for example, if I'm folding forward to pick something up off the floor, the body spontaneously takes a breath in, but there's an expansion quality.
Yeah.
Right. Whereas in yoga, we were always taught to exhale and fold forwards, but then that becomes, it's not efficient. It doesn't feel whole. So I'm just sharing what these movements that emerge are informing me, that this auxetic nature of the tissue, where does it happen in the body? I don't know either, but all I can share is that the body feels springier. It's like there's a kinetic energy that's restored, and that there might be days where I feel like I want to run. I'm not a runner, but there's days where I feel, I feel like I want to run. But in that running, it's more spring-like than it used to be. And it's from this
such
auxetic nature of the tissue.
Yeah. So, I also not only in terms of biomechanics, but in other dimensions, you could call the regular behavior of the IT band that gets narrow when you stretch it. That would be focused movement. So if you go in one direction, you sacrifice another one. That's what nature sometimes needs to do. So you say, this month I will focus on this, and I will have less time for another important dimension in my life. So you say, I'm going to study for my exam, and you don't greet your wife when you have breakfast with her. No, I'm focused there, yeah. And you run through the streets to go jogging, and you don't look to the right and left, and you're very focused there. And nature is doing that sometimes. So you invest in one thing, and you need to sacrifice. Everything has a trade-off. So that is one there. So you focus on the expense of other dimensions. But there are also processes where it's going in multiple directions. So you go jogging, but you also take more in the colors of the trees and your neighbor, and you may smile to them. So you're not cutting down there.
Yes.
And that for us is almost a richer sense, because you don't have constant volume, you are expanding. So the more you go upwards, the more you also spread to the right and left and to the back and to the front. And it's a nicer movement, if you ask me.
Oh gosh, it feels pleasurable, and it's also it happens from within.
Yeah.
It's just this sensation, the urge to move.
Yeah.
Happens from this area of the diaphragm, the heart. It's just
and you are in a much more interesting. So sometimes I need to focus. You know, I need my work done for tomorrow, and I become very German and metallic and masculine, and it's not nice for me. But it's often the only way how I get a job done. But then if I can change into multi-directional organization, it is much nicer. So I prefer the second way.
Yeah, and I hear you. We're kind of in the same, you probably more than me, because I can dip in and out. I mean, Robert, you definitely more than me, because I'm much more, I'll just do what I can, you know. I want to do this certification, but how much effort is it taking?
Yeah.
And sometimes we may need to go there because we want to share this work.
Sure.
But there's most of the time, the way we're meant to live, which is what we're trying to help people with, is to find this wholeness within their being, and how to restore that sense of wholeness. What Jaap van der Waal talks about, wholeness, you know, how do we reconnect with that? In the somatic world, we call it soma, whatever we call that. So it's bringing this awareness of our sensory nature. So I'm kind of coming, because I know I've taken up a lot of your time already, coming to that sensory nature, and respecting how the body is designed, how we can use it for clues that emerge from the body. So if we have any stiffness, we have a better understanding of why, and it has more to do with our emotional state. It might be with the fact that we've been sitting for a long period of time. So it gives us that information. But what it's also given us today is how you can shift that, and the way that we do it in intuitive practice is the spontaneity, the coaxing, the feel-good movements that emerged from finding that.
So the Auxetic one feels much better, you know. So if you are dancing, and you're not just going like this, Heil Hitler, you know, and you don't look right and left, but if you go like this, and you still are aware of the exchange in multiple directions, it is much nicer.
I love that. I love that multiple direction, multi-dimensional. Gosh, Robert, I think we need to have another session. There's just, it's so rich, you know. This one session is never enough. So I may need to get you back and get talking more about this. So it's been amazing, and just to reconnect on a one-to-one with you today was really special. So thank you. I'm just going to